My name is Lexus Castor, and this is the prokaryotic replication fork. As we see here in blue is going to be the origin of replication. An indicator protein will come along, recognize the origin of replication, bind to it, and initiate the DNA unwinding. Enzymes to help with unwinding, we have here helicase, which is going to break the hydrogen bonds between the base pairs of double-stranded DNA, which will break the helix apart. To keep the DNA single-stranded, we have single-stranded binding proteins seen by this rice. They're going to coat the DNA and prevent the formation of secondary structures. Lastly, we have gyrase by these red circles that are going to relieve the torsional strain in front of the replication fork. Without gyrase, the DNA would remain supercoiled and replication would not be able to occur. To begin actual replication, you're going to have RNA primase that comes by and lays RNA primers, shown here by yellow, on each strand. To create DNA nucleotides from the RNA primers, you have DNA polymerase 3 that will add DNA nucleotides in the direction of 5' to 3'. The template strand here is 5' to 3' this way, so the newly synthesized DNA will be 5' to 3' this way. Since yellow and pink make orange, I'm just going to go ahead and add this newly synthesized DNA, and it will continue continuously in this direction as this fork is opening, and that is why we call this the leading strand. On the bottom, it's going to be the opposite direction. The template strand is 3' to 5', which means replication will occur 5' to 3' this way. This ultimately creates fragmented DNA, which we call Okazaki fragments. And this is the lagging strand. Shown below here is another way to visualize it. This is actually how it occurs with like a Y-like structure with looping DNA. The DNA polymerase 3 are going to be dimerized, and the replication will continue as I mentioned up here. Now the replication has completed, we still have these Okazaki fragments and we need to get rid of them. So to do that, we have DNA polymerase 1 that will eliminate the RNA primers and replace them with DNA nucleotides. It will do that for the entire strand until we have all DNA nucleotides. Now all we have left is this gap in the DNA nucleotides, which is a missing phosphodiester bond, which is what they call a NIC. The enzyme ligase will come by, combine these two strands by adding a phosphodiester bond, and replication is complete.